Learning goals. Welcome to this learning session on limiting factors in photosynthesis. In this course, we shall address Introduction to Limiting Factors in Photosynthesis, Light Intensity, Investigation and Graph Analysis, Carbon dioxide concentration and its effects, Temperature, Limiting Factor and Denaturation, Interactions and Optimizing Crop Yield. There will be a quiz at the end to test your knowledge. Introduction to limiting factors. Limiting factors are environmental conditions that restrict the rate of a process, even if other conditions are optimal. In photosynthesis, these factors prevent the plant from performing at its maximum potential, much like the slowest worker on an assembly line dictates the overall production speed. The primary limiting factors for photosynthesis include low temperature, dim light, and insufficient carbon dioxide concentration. Understanding these factors is crucial for maximizing plant growth in both natural and controlled environments, such as greenhouses. The photosynthesis equation. Photosynthesis is the process by which green plants and some other organisms use sunlight to synthesize foods with carbon dioxide and water. This fundamental biological process converts light energy into chemical energy, stored in glucose. The overall chemical equation for photosynthesis demonstrates the inputs and outputs, carbon dioxide and water, in the presence of light energy, yield glucose and oxygen. This reaction is vital for nearly all life on Earth, as it produces the oxygen we breathe and the organic compounds that form the base of most food webs. Light intensity as a limiting factor. Light intensity directly influences the rate of photosynthesis because light provides the energy needed for the reaction. At low light levels, the rate of photosynthesis is directly proportional to the light intensity, meaning more light leads to a faster rate. However, there is a saturation point where increasing light intensity further will no longer increase the rate of photosynthesis. This occurs because another factor, such as carbon dioxide concentration or temperature, has become the new limiting factor. For instance, in deep ocean environments, light intensity is the primary limiting factor, leading to unique adaptations in photosynthetic organisms. Investigating light intensity, pondweed experiment. The effect of light intensity on photosynthesis can be measured by observing the rate at which oxygen is produced by a plant, such as pondweed. Pondweed is ideal because it visibly releases oxygen bubbles when photosynthesizing underwater. The experimental setup involves placing pondweed in a test tube filled with water, inverted over a funnel, and collecting the oxygen bubbles in a measuring cylinder or another inverted test tube. By varying the distance of a light source from the pondweed, the light intensity can be controlled, allowing for precise measurement of oxygen production over time. This method is a classic demonstration of gas exchange in plants. The graph illustrating the rate of photosynthesis against light intensity typically shows an initial linear increase. At this stage, represented by point A on the graph, light intensity is the primary limiting factor. This means that as light intensity increases, more light energy is available for the light-dependent reactions of photosynthesis, directly accelerating the overall process. Every additional unit of light leads to a proportional increase in oxygen production, indicating a direct relationship between light availability and photosynthetic rate. Analyzing light intensity graph. Plateau phase beyond a certain point, the graph plateaus, as shown by point B. Here, increasing the light intensity no longer increases the rate of photosynthesis. This indicates that light is no longer the limiting factor. Instead, another factor, such as carbon dioxide concentration or temperature, has become the limiting factor. The plant is now utilizing all available light energy, and its rate of photosynthesis is capped by the scarcity of CO2 or the suboptimal temperature. This phenomenon is known as light saturation, a critical concept in agricultural science for optimizing crop yields. Carbon dioxide concentration. Carbon dioxide is a crucial reactant in photosynthesis, specifically in the light-independent reactions, Calvin cycle, where it is fixed to produce sugars. Therefore, its concentration directly impacts the rate of sugar production. At low CO2 levels, increasing the concentration of carbon dioxide will directly increase the rate of photosynthesis, as more raw material is available for the plant to convert into glucose. This is why commercial greenhouses often use CO2 enrichment systems to boost crop yields, sometimes increasing CO2 levels up to 1000 ppm, significantly higher than the atmospheric average of around 420 ppm. Carbon dioxide concentration graph. Similar to light intensity, the relationship between carbon dioxide concentration and the rate of photosynthesis is initially linear. As CO2 concentration increases, the rate of photosynthesis rises proportionally. However, once the CO2 concentration reaches a certain level, the graph plateaus. At this point, carbon dioxide is no longer the limiting factor. Instead, light intensity or temperature has become the limiting factor. This saturation point highlights that simply adding more CO2 will not indefinitely increase the photosynthetic rate if other essential conditions are not met. Temperature as a limiting factor. Temperature significantly affects the rate of photosynthesis because the process involves numerous enzyme-catalyzed reactions. Enzymes are biological catalysts that speed up chemical reactions, and their activity is highly sensitive to temperature. As temperature increases, the kinetic energy of molecules also increases, leading to more frequent and energetic collisions between enzyme and substrate molecules. This generally accelerates the rate of enzyme activity and, consequently, the rate of photosynthesis up to an optimal temperature. For example, the enzyme rubeo, crucial for carbon fixation, has an optimal temperature range, outside of which its efficiency drops. Temperature graph and enzyme denaturation. The graph of photosynthesis rate versus temperature typically shows a bell-shaped curve. The rate increases with temperature up to an optimum point, reflecting enhanced enzyme activity. However, if the temperature becomes too high, enzymes begin to denature. Denaturation is the irreversible change in an enzyme's three-dimensional structure, causing it to lose its active site and thus its ability to catalyze reactions. This leads to a sharp decline in the rate of photosynthesis, as the essential enzymes become non-functional. For instance, most plant enzymes denature above 40 to 45 degrees, causing significant damage to the plant's metabolic processes. Interactions of limiting factors. It is important to remember that only one factor can be limiting at any given time. If light intensity is low, increasing carbon dioxide or temperature will not increase the rate of photosynthesis until light is no longer the limiting factor. The concept of limiting factors is dynamic. As one factor is optimized, another may become limiting. For example, in a greenhouse, if light is increased, the plant might then become limited by CO2. Once CO2 is also increased, temperature might become the next limiting factor. This interplay requires careful management in controlled agricultural settings to achieve maximum efficiency. Optimizing photosynthesis for crop yield. Understanding limiting factors is paramount in agriculture, particularly in controlled environments like greenhouses. By manipulating light, CO2, and temperature, farmers can create optimal conditions to maximize the photosynthetic rate and, consequently, crop yield. This optimization involves balancing the cost of providing these resources against the increased revenue from higher yields. For example, supplemental lighting, CO2 enrichment, and precise temperature control are common practices. Advanced techniques such as hydroponics and aeroponics further enhance nutrient delivery allowing plants to fully exploit optimized environmental conditions for rapid growth and increased productivity.
Summary. Well done, you have completed the course Limiting Factors in Photosynthesis. You should now be familiar with Introduction to Limiting Factors and Photosynthesis, Light Intensity, Investigation and Graph Analysis, Carbon dioxide concentration and its effects, Temperature, Limiting Factor and Denaturation, Interactions and Optimizing Crop Yield. Should you need to revisit any of the topics in this course, please use the menu at the bottom of the screen.